is gametogenesis, the process of formation of gametes. Now, first is spermatogenesis. The formation of sperm is known as spermatogenesis, and this is actually the male gamete. So, okay. So there are two steps for the formation of spermatogenesis or for the formation of mature sperm. First step is formation of spermatid, and the second step is formation of spermatozoa. We have divided this into few phases, three phases. Okay. Number one is multiplication phase. Let's remember where it is going to happen. It will occur in the seminiferous tubule. Suppose this is the lining of seminiferous tubule. These are germ cell. These germ cell will produce large number of spermatogonia. Polarity of germ cell is 2n. Polarity of spermatogonia is also 2n. So during the multiplication phase, the spermatogonia multiply to produce primary spermatocyte. The division here was mitosis. So the polarity of primary spermatocyte will be
2n.
2m. Clear? So what happens here? The spermatogonia enlarge in size and form, multiply and form primary spermatocyte. This, as soon as this primary spermatocyte form, just the second phase starts growth phase, and in the growth phase, the primary spermatocyte will divide. There will be first meiosis, and it will lead to the formation of secondary spermatocyte. Now the polarity of secondary spermatocyte will be haploid. So from here to here, till the first division started, before the first division started, there is a phase known as growth phase. But as soon as the secondary spermatocyte is formed by the primary spermatocyte, the phase will be called as maturation phase. Okay, so in this phase, the secondary spermatocyte undergo one more meiotic division and produce 4 haploid cell, and these haploid cell will be known as spermatid. The second one is formation of spermatozoa. Okay. Now actually this is nothing but the transformation of spermatid, into spermatozoa. What actually happened in this stage? First of all, formation of acrosome. Okay, and this acrosome is formed by Golgi body. Formation of axial filament, and this axial filament is formed by distal centriole. Elongation and condensation of nucleus. Spiral shape mitochondria coil around axial filament in the middle piece. And lastly, formation of flagellum. Okay, when all these things completes, we can say it is the spermatozoa. Okay, so let us see the structure of sperm. So when we look into the structure of sperm, we see how many portion: head, middle piece, and tail. Somewhere neck is also mentioned. So what we see in the head portion, we see first of all head portion is consists of a membrane known as plasma membrane. Okay, this head portion consists of two most important thing. Number one, a sac-like structure consisting of हाइड्रोलिटिक एंजाइम एंड लाइटिक एंजाइम जैसे हाइलोरोनिडेज है, न्यूरामिनिडेज एंड zonal lysin. The nucleus will elongate. The nucleus will elongate and condense, and it will consist of the chromosome. Clear? One more thing, when the spermatid transform into the spermatozoa, the centriole separate. This one is proximal centriole, distal. Okay, now if you remember, the distal centriole is playing role in the formation of axial filament. What is the role of proximal centriole? Cleavage. It participate in cleavage. Whenever the first cleavage occur, it will participate. As soon as the neck finishes, the another structure starts known as middle piece. Okay, the middle piece consists of centriole and axial filament and also ribbon shaped mitochondria. And this mitochondria is also called as Nabon kernel. So the last portion of the sperm is tail. Basically, it is for the movement. What is the role of mitochondria here? It provides energy for the movement. What we can see in the neck portion, we can see that only proximal centriole is present in the neck portion. You must remember that middle piece consists of centriole and mitochondria, whether centriole is distal centriole. So what will happen to the mature spermatozoa after maturation? Head, this is the head now. Okay, this head will embed into the Sertoli cell for the nourishment. Okay, and when they are fully nourished, they are released from the seminiferous tubule, and this process is known as spermiation. Okay, now what will happen to this mature sperm? Where will they get deposited? Epididymis and early vas deferens, where they get nutrition, physiological maturation, fertilizing capacity, and all. Okay, all these things, the formation of sperm take 64 days. So in the epididymis, the sperm get stored for whole as well as in vas deferens, whole 1 month. Okay, and if they are not ejaculated, they will be reabsorbed.